Hello everyone, welcome to ITLS Academy. This is your mentor Dr. Shamaita Basu, and in this session we will be starting with a new topic of this course of forensic microbiology. So as we can see over here, the topic or the module that we will be starting today is sampling methods and data generation. So again, I think this will be taking at least 2 or 3 sessions to complete this topic. So today we will be focusing on the first part of this topic of sampling methods and data generation. Let's move on. So again, first focusing on the introductory part, that is what do we actually mean by sampling methods and why, what is the importance or what is the place that this sampling methods is holding uh, in this field of forensic microbiology. So let's see what they're saying over here. The study of forensic microbiology is an inherent blend of forensic science and microbiology, and both disciplines have recently been undergoing rapid advancements in technology that are allowing for exciting new research avenues. However, the integration of two different disciplines poses challenges because experts in two different fields are required to become competent, if not proficient, in the conceptual and technical background and tools for sampling and data generation of the other discipline. So basically what they're saying is that to make this thing work out, means make this sampling collection technique work out in the field of forensic microbiology, it is a bit of challenging situation, or it is a bit of a challenge. Why? Because we need to take two individuals, or we need to hire two individuals which are actually expert in two different fields, one in the field of data collection and surveys and the other in the field of forensic microbiology, obviously. Okay, so basically to deal with or to put two persons belonging from two completely different fields together, make them work together, make them cooperate together is a big challenge, obviously, because they're totally belonging from two other fields. Okay, so one will not understand the the struggles or the job roles or the the work environment of the other person, and vice versa. Okay, so that's why it's a challenge. For instance, most microbiologists have not been trained in how to approach death scenes or interpret data within a forensic context. Likewise, forensic scientists often have not received in-depth training in classical microbiology or the recent molecular advances that are somewhat revolutionizing these fields. So, just as I mentioned, that the person who is belonging from the field of forensic science will not be able to understand the data interpretation, the sample collection, the taking survey part of the other person. And similarly, the other person will not understand the details about the molecular diagnostics, the crime scene investigation, these things, obviously, the integral part of these things, the deeper parts of these things, of these aspects, obviously that data collector will not be able to understand. So therefore, why we are talking about this particular chapter, sample collection, because it will be providing an introduction to sampling methods and data generation that are an integration of techniques from laboratory based microbiology, field and environmental microbial ecology, and considerations within a forensic context. Okay, now firstly we will be talking about financial considerations. See, again, since we are talking about forensic microbiology mainly with focus in India, right? And this is something I say again and again, like, this is unfortunate, but this is the truth, that still as India we are developing, obviously we are developing, we are flourishing in many aspects, but still we are a developing country. We have not we have not yet reached the tag or the stage of a developed country. We are still a developing country and a poor country and a third world country. Okay, So financial restrictions, financial limitations are a big thing, big issue that we face whenever we talk about, science progression, technological progression, research progression, medical science progression, even engineering science progression. All these things, they take a backseat, or we face a setback as the scientists or the researchers because of fund issues, because of financial issues, financial constraints, and also not only lack of fund but also plenty of corruption, corruption at every level, like from the level of the ministers to the level of the the security guards. Each and every level we are having corruption, as a result of which whatever funds we are having, already we are having low funds because we are a third world country or developing country, and on top of that most of those funds are going in vain for this corruption thing. So ultimately हमें सिर्फ चिल्लर मिलता है. When the main work is getting done, the fund that is remaining is very very less, almost negligible. So that's why we are always lacking behind. So this is a huge constraint that we are facing, financial constraint or fund constraint. Okay, so let's see. Often one of the most important constraints to any research or application of a given discipline is financing, and forensic microbiology is no exception. Financial limitations will often dictate the experimental design of most research projects. The number of replicates, the number of samples, and how often they are collected in a study all require consideration for the cost of each sample, in addition to other expenses necessary to successfully complete a project. Example, high throughput sequencing or server space. While the cost of the expandable supplies, for example swabs or DNA extraction reagents, and service activities such as various forms of genomic sequencing or purchasing specific bacterial cultures, are often considered early on in the development of research, the costs associated with personnel and time are often miscalculated. Therefore, a realistic estimate of the costs associated with acquiring permanent equipments, expandable supplies, contractual services, personnel, and other costs necessary to undertake the research are critical for successful research projects that reflect a rigorous and scientifically defendable experimental design. Okay, moving forward. Microbial research has historically been expensive, and new advances in genomic technology have continued to be costly, especially when the research is new to a scientist without funding. So this is again a very important point, that whenever see, just as many people or many personnel are hired as freshers in different companies, right? IT companies or tech companies. Similarly, in the research field also, some of the other scientists will be a fresher, right? Like he or she will be new in that field of science, new in that field of scientific research. So for them to be, to be specific about how to utilize the fund, because again the same thing, whatever fund we are getting is already less than what we need. So we are already lacking in the first place. On top of that, the person who is actually concerned, the person who is actually responsible to to disburse those funds or to distribute those funds in the right places, if that person is not that much experienced, if the person himself is confused, if the person himself is new to the field, which can obviously happen, it's not something abnormal or it's not something that should not happen, but it becomes more challenging for that person, it becomes a more difficult situation to ensure that the funds will be used properly in proper sections. In that case, it's not that the scientist is corrupt, I'm not saying that the scientist will be utilizing the money for his or her own favors or own family members. No, it's not like that, though there are cases like that also. in government sectors and all, particularly in government universities, in government research institutes. I myself have seen such examples where some project has been approved, maybe of ICMR or ICAR, whatever. Some project has been approved and that person has become the PI, that is the principal investigator of that project. So he or she is like hiring scholars and telling them that, okay, I will be paying you this much of fellowship, you have to work for me, you have to work for this project. But actually, maybe, like, as the PI, he or she is ought to pay that scholar maybe 15,000. But, like, according to the, when a project is sanctioned, when a project is approved under a particular scientist, like, if I am the scientist, then I have to write the project. I have to write what I'm going to do with the project, what I'm going to, like, what will be the end product, or what will be the outcome of my project, what are the different steps, what are the different methodologies that I need to follow, what are the different uh, you know prerequisites that I need. And on top of that, I also need to mention the manpower, that, okay, for to make this project successful, I need this much of lab technicians, I need this much of research scholars, this much of research fellows, or this much of research assistants, or whatever it is. The post varies according to the the budget of the project, or to which body I'm submitting the project, whether it's ICMR or ICAR. Depending on that, the name of the positions vary, but the job is the same, that of a lab technician or that of a scientist. These are the two main posts that we need. So the positions, the number of posts also I will give as the the author of the project, and their their fellowship also I will decide. But I have a scale, I have to decide on that range only. Say for example, it's between 10 to 20,000, so I have to give within that range only. Okay, I cannot make it 50,000 or I cannot make it 5,000. So the example that I was talking about, that particular PI, she had you know given herself given the budget as 15,000. Maybe the range was between that only, so she had to give 15,000. Like, she had to show that I will be paying my scholar or my fellow, research fellow, this amount of fellowship per month. But when actually she hired a scholar for that purpose, that scholar was also a girl, she told her that I will be able to give you 9,000 only. And what she did with the rest of the money, like 15,000 ke jagah, she was giving 9,000. So per month she was saving 6,000, right? And with that money, she took a AC for her own room, for a staff room. She took a AC and she put up the AC. So this is the level of corruption. Just as I mentioned, that the corruption begins from the level of the ministers to the security guards. So each and every level we have corruption. So that is another example. But what they're saying over here is that if the researcher is new, if the researcher is a fresher, then it becomes a difficult scenario. Okay. This is particularly true in situation exactly what I said, which is often the circumstance for many new researchers in forensic microbiology. Additionally, to best represent real world scenarios and natural circumstances that are relevant to forensics, researches in many aspects of forensic microbiology are conducted in the field rather than in the laboratory. Moving such research from controlled laboratory conditions into more realistic settings poses new and sometimes unexpected costs to the research budget. For instance, a laboratory microbiologist may be aware but may not anticipate the cost associated with doing field research on multiple human cadavers, such as the equipment, example vehicle, to transport the cadavers, or the environmental instrumentation necessary for measuring and monitoring weather conditions, or the material and supply costs for building cages to go around the cadavers to prevent undesired scavenging events by the vertebrates. Okay, now if we talk about terrestrial settings, firstly under this we need to talk about the environmental conditions. There are six primary terrestrial biomes. They are tundra, taiga, temperate forest, desert, grassland, mangroves, and tropical rainforest. that can be divided into 14 major habitat types with 867 smaller ecoregions according to the World Wildlife Fund. Each ecosystem has unique and important biological functions such as nutrient cycling, but also has various abiotic characteristics like temperature, pH, terrain, and plant diversity. The variation in biotic and abiotic diversity encountered across ecoregions may present challenges that forensic practitioners must consider when assessing naturally occurring microbial communities associated with decomposition. For example, in North America, there are 15 level 1 terrestrial ecoregions ranging from the Arctic cordillera in northern Canada to tropical humid forests in the lower parts of Florida and southern Gulf coasts of Mexico, and including various plains, mountains, deserts, and forests ecoregions distributed throughout the continent. Commission for Environmental Cooperation Working Group 1997. Next, cadavers can be discovered in almost any terrestrial location, that is biomes, under numerous circumstances that can either completely conceal the remains by burial or leave the remains fully or partially exposed. Thus, researchers conducting decomposition studies to characterize microbial communities after death should document abiotic conditions throughout the duration of an experiment, since temperature, pH, and other physicochemical properties influence microbial community dynamics from an ecosystem to microhabitat scale. There are numerous instruments available to measure abiotic conditions, but as previously discussed, the financial circumstances will be important for frequency and accuracy of data collected. Local meteorological conditions like temperature, humidity, and rainfall are important abiotic factors that can influence the rate of decomposition and the development of consumers feeding on the remains. Example, Caliphodire, which is Diptera larva. Next, we will be talking about consumer collections. Many consumers utilize and interact with decomposing remains for food, shelter, progeny rearing, and or mating location. There are many excellent texts available that discuss in depth the role and community dynamics of invertebrates and vertebrates during decomposition. Primarily, insects and other vertebrates have the most contact with decomposing remains and therefore may be of the most interest for assessing their microbiology. Files can be mechanical vectors of numerous microorganisms. Flies can be mechanical vectors of numerous microorganisms like bacteria, fungi, archaea, and protists. They can inoculate the remains with microbes from other sources of decomposing organic material like manure found at a livestock facility and can acquire microbes from the remains of potential distribution back into the ecosystem. Additionally, specific species of blowflies like Diptera and beetles like Coleoptera can produce antimicrobial peptides which can ultimately alter the microbial community composition on the resource. Continuing, there are several entomological trapping methods widely used depending on the species of interest to be collected. Examples of these methods include but are not limited to flight intercept trap, malaise trap, light trap, beating sheet, sifting, which is also known as burlesque funnel, and carbon dioxide trap. Forensic entomologists typically use a subset of methods for collecting the insect communities. Flying insects commonly associated with decomposing remains such as blowflies and flesh flies, which belongs to Diptera sarcophagidae, can be collected either using active trapping methods like sweep nets or through passive trapping methods like baited inverted cone traps or glue traps. Gloves should be worn when collecting samples in order to minimize cross-contamination and exposure to potential pathogens. Insect samples can be placed in sterile glass vials, conical tubes, or microcentrifuge tubes, depending on the number of specimens collected. Glue traps can be carefully collected, closed cylindrically with the sticky side facing the interior to protect the insects and prevent collapse, and then placed in polyethylene bags. Litter or soil samples also can be stored in sterile glass vials, conical tubes, microcentrifuge tubes, or polyethylene bags. So basically, this is an example that is given of these different polyethylene bags and different examples of this collection of jars and all, regarding this collection. Okay, so in this image, in the first image, that is A, left hand side top, it is showing a inverted cone trap for collecting insects. A bait resource, in this instance, the white arrow pointing to a stink jar. So this is the white arrow you can see over here, and this is the stink jar, consisting of decomposed liver and water, and it is placed at the bottom opening as an attractant for necrophagous insects. So since it's having the liver, that is the rotten liver, which is the decomposed part of a animal carcass or human carcass, so that will be attracting all the necrophagous insects. Why necrophagous? Because they're feeding on this necrotic tissue, that is the liver. This one is B. Sorry, this is B. Right top. Okay, it's showing an anti-scavenging cage placed over a swine carcass. So see, this is the swine carcass that is there. The field is an excellent place to attach passive trapping methods such as glue traps. This is the glue trap that is seen over here. Okay, see, glue is placed, so on the glue the insects are coming and getting stuck. For collecting flying invertebrates attracted to carrion. Carrion means the decomposing material or the carcass. D, second last, left hand side below, lower left. Swabs can be effectively used to collect the epinecrotic microbial communities from cadavers. So see, this is basically a human cadaver from where the sample is getting collected by this lady. And lastly, right bottom we have again jars. So this is basically showing an aquatic habitat. So in aquatic habitats, stable structures placed in an aqueous habitat, such as these unglazed ceramic tiles. So these are ceramic tiles that are shown over here, attached to a brick. So this is the brick, obviously you can understand. Create a standardized area for biofilm formation and can be left for weeks to months or up to a year depending on the scope of the study. Now what do we mean by biofilm formation? It's basically when too many bacterial colonies are grown at a particular place or at a particular location, and it's so huge in number that it produces a thick layer. And that what do we mean by film? Film means a layer, right? So many colonies grow together that they produce a thick layer, and that thick layer is known as biofilm. Okay. Okay, now if we talk about aquatic settings, they can be generally grouped into freshwater and marine, but there is some interference of these habitats like brackish habitats in estuaries and some saltwater inland lakes. In addition, these habitats can be flowing, relatively stagnant, or not flowing. Flowing aquatic habitats are streams, rivers, large lakes, or oceanic currents, while no or little flowing habitats are ephemeral ponds, wetlands, marshes, and lakes. Ephemeral means very small. Some parts of oceans have little flow, but most of the oceans and large lakes have dynamic water movement in the form of vertical and horizontal currents. These habitats, whether flowing or not, are functionally important to microbial communities because nutrient availability, oxygen content, ion concentration, sedimentation, and many other variables are substantially different in aquatic environments. To account for these abiotic variables, there are numerous materials and instruments that are necessary and different from those used in terrestrial settings. When cadavers are discovered or studied in aquatic settings, there are several water quality variables that should be measured because they are important factors to decomposition, especially microbial communities. These water quality condition variables are discussed in more detail elsewhere, but the primary factors can be measured either as spot, one-time measurements, or unattended with continuously measured intervals, for example 30 minutes, 1 hour, and 24 hours are the following: dissolved oxygen, temperature, pH, redox potential, various ions like chloride and iron, conductivity, turbidity, total dissolved solids, clarity, and suspended solids. Additionally, many of these variables can change over small areas, or so it is important to note where the water quality parameters are measured. Stream flow velocity is known to affect how microbial community biofilms form on decomposing carrion in streams along with other variables like oxygen, temperature, and sedimentation that affect the aquatic biofilms. There are multi-parameter probes that are capable of measuring several water condition variables simultaneously, either for a single spot assessment or with the capability of unattended sampling. These probes are often quite expensive, for example over $15,000 for the housing unit, all probes and connecting cables, and can often be cost prohibitive. There are also smaller, more cost-effective probes that do not have the attended sampling capability and often can only measure 3 to 4 water quality condition variables, which are usually dissolved oxygen, temperature, conductivity, pH. These probes are often a suitable yet limited alternative to multi-parameter unattended probes for aquatic research. Further, if the microbial communities of the water column are an important research question, there are several potential avenues to filter the microbial and associated sediments into filters, measuring the volume of water that is filtered and matching this to water quality condition. These filtering strategies can range from small handheld syringes to larger battery-powered vacuum pumps to automated stormwater samplers that pump water from the source and then store the water in climate-controlled conditions for later filtering. Now talking about sample collection techniques. The use of proper aseptic sterile techniques to eliminate contamination of collected microbial samples that will undergo sequencing analysis is the most vital protocol considered in any experiment. As microbial forensic techniques are developed, it is imperative that the collection of samples for future forensic DNA or RNA analysis be efficient and non-destructive, and that contamination by surroundings or personnel is mitigated. Inadvertent contamination of a sample with extraneous microbial DNA or RNA is unfortunately quite possible given the environment in which samples are taken, and if it goes undetected, contamination sequencing reads will be incorrectly identified as legitimate results. Therefore, aseptic sample collection techniques should always be taken seriously, and extra caution is always warranted. All materials that will come into contact with the sample must be sterile, and manipulations of the sample must be designed to avoid contact with any non-sterile surroundings. Personnel collecting samples should be required to wear PPE such as booties, gloves, aprons or suits, sleeves, safety glasses, surgical masks and hair covers. PPE will prevent both contamination of sample with extraneous microbes and possible passage of infectious pathogens to personnel. However, just donning PPE does not automatically prevent contamination of a sample. It only acts as a sterility barrier. It is up to the person wearing the PPE to be purposefully aware of their actions in order to avoid contact with non-sterile environments or prevent contamination of themselves. An absolute sterility barrier cannot be accomplished in the field without prohibitively impeding routine manipulations and sample collections. Therefore, procedures should be designed to significantly reduce the probability of contamination. One primary consideration is to arrange your supplies within your work area to be easily accessible with wide spacing so that you avoid reaching over one sample to get to another. If items in use are tightly packed, you will inevitably brush a sterile surface with a non-sterile one or inadvertently drop non-sterile items, for example dust, skin cells and dirt, into sterile containers. Whenever possible, hold items such as collection tubes and swabs in your sterility gloved hands or sterile tube holders and avoid setting them onto non-sterile surfaces. Next, sample preservation, storage and handling techniques. A sample must be taken in such a fashion that it is representative of the microbial community. For example, skin microbes from healthy humans can usually be collected by taking swabs, scrapes or punch biopsies. Each of these collection methods has pros and cons as far as convenience, cost and applicability in the field are concerned. From a forensic perspective, minimal disturbance of evidence is desired. Therefore, non-invasive methods such as swabbing with a sterile cotton swab or forensic collection swabs made from rayon, polyester, or foam are preferred. As the number of microbes on the epidermal surface may be low, special care must be taken to adequately sample in order to capture a usable quantity of microbes that assures sufficient high purity DNA for PCR and subsequent sequencing. This is determined by systematic experimentation where all sampling is standardized in terms of the type of swab used, the time spent swabbing, for example 30 seconds and 1 minute, and the size and location of the area swabbed. Microbes are generally present in large quantities in the GI and GI tract and body orifices like buccal cavity, that is the oral cavity, making collection from those areas easier. In general, 0.25 to zero point 0.5 gram of microbial GI matter material per sample is sufficient for DNA isolation. Replicate samples should always be collected if possible, and not just the minimal triplicate to assure statistical relevance, but also additional samples for possible replacement of corrupted or low yield samples. Sterility factors and temperature need to be controlled and standardized for field collection. Dependent on the experimental design, the amount of time spent collecting samples prior to processing in the lab could be significant and enough to affect microbial species viability. Therefore, immediate short-term sample storage in the field, such as placing samples on ice packs in a cooler, should be a consideration in the experimental design. So I think that's it in this session. We will be soon moving on to the next session regarding this topic. Till then, thank you and goodbye. Please like, share and subscribe ITLS Academy.